Hello there, and thank you for tuning in to see what the world looks like through atheist eyes. I'm Frank Zindler from American Atheist Press, and I have been your host for programs on a wide variety of subjects. Today's program is the fourth installment on what I believe will be the most important programs that I shall ever produce. These programs deal with the causes of aging and what science is learning about how to forestall those processes and indeed even how to reverse them. In a sense, I have become your escort and guide in a quest for the fountain of youth. I've already shown how religion has retarded and even blocked immortality research. I've shown you some evidence that aging and lifespans are genetically determined. Today, we shall get down to some specifics of how genes determine lifespan. Today's program is called Methuselah's Genes. Since I tried, back in the 70s, to coin the term opsiphonial genes to denote genes that determine when and how a living organism should be shut down and die, there has been an enormous growth in research on the genetics of aging and the determination of lifespans. According to the database called GenAge, at this time, there are over 800 genes known to extend lifespan in model organisms. That includes 454 genes in the simple nematode worm, Caenorhabditis elegans, that's also pronounced Caenorhabditis by some people, 236 in the yeast Saccharomyces cerevisiae, that's what you use to brew your beer, 79 in the fruit fly Drosophila melanogaster, and 68 in the mouse, Mus musculus. In the case of the roundworm, one of the earliest animals in which aging-associated genes were discovered, it was shown by Cynthia Kenyon that a mutation in a gene she called DAF2, DAF2 stands for decay accelerating factor 2. Even these goofy names that they come up with for all these genes and proteins, they actually are supposed to be meaning something. She was able to double the lifespan of the worm. I was present at the meeting of the American Association for the Advancement of Science when she read her paper. And I was so excited, I could hardly speak to ask her a question after her presentation. Later, it was shown that modification of another C. elegans gene, PI3K, could increase maximum lifespan, are you ready? 10-fold. Wow! If you had had enough time to think about what I've already told you, you might have asked me, if a single gene could extend lifespan 10-fold, then why are there 453 other genes involved in determining the lifespan of this worm? Would their effects all be additive? If we appropriately mutated all 454 known longevity genes, could we get a nematode to live to be more than 1,000 times older than normal? Could that be extended to humans? By now, you already must have suspected that the subject of how genes control aging must be very complicated, and that it will take a number of programs later in this series to adequately explain that. So I need to talk about something right now that is, although it's unrelated to aging specifically, it can clarify the nature of our problem in the genetics of aging, because it is logically analogous to the problem we are facing. This will set the stage for understanding aging. Let us go back in time to February 17th, 1600, to the burning at the stake of Giordano Bruno, the martyr for science, who suggested that the stars were distant suns and that they too must have planets going around them. let us ask the seemingly simple question, who burnt Bruno at the stake? Who or what was the cause of his incineration? Of course, you might answer that it was the unidentified guy who touched the torch to the pyre. But of course, if some other unidentified guys hadn't piled up the combustible materials for the pyre, nothing more than Bruno's clothing and hair would have been burnt. Okay, so Bruno was burnt by a combination of a torch-wielding henchman and one or two lackeys who piled up wood. Does that satisfactorily answer the question, who burnt Bruno? I think not. You might argue that it was the Holy Roman Inquisition that burned him, due to the fact that it was the Holy Roman Inquisition that condemned him to death. But remember, it was the Venetian Inquisition that initially had him arrested for heresy. And then Rome pulled rank for the privilege of murdering him. All right, so it may have been the Holy Roman Inquisition that burnt Bruno, but who exactly was the Holy Roman Inquisition? Of course, it was a group of highly religious men, deeply devoted to gentle Jesus. The Grand Inquisitor was a Jesuit cardinal saint, Robert Bellarmine. Incidentally, Bellarmine was made a saint on the 29th of June, 1930, by Pope Pius XI. That was the guy who helped Hitler come to power. Certainly, if Bellarmine hadn't wanted Bruno to be converted into carbon dioxide and water after he had been unable to convert him to orthodoxy, he wouldn't have been burnt. But Bellarmine needed the votes of the entire Inquisition to make the murder a sacred act. So who else was on the panel? Well, there was Cardinal Camillo Borghese, later to become Pope Paul V. There was Cardinal Paolo Emilio Sfondrati, Cardinal Badruzzo, otherwise called Madruzzi, Domenico Cardinal Pinelli, Pompeo Cardinal Arrigoni, Pedro Cardinal de Deza Manuel, and Cardinal Santorio, the Archbishop of Santa Severina and the Cardinal Bishop of Palestrina. Wow. So, was it the entire Holy Roman Inquisition that burnt Bruno when it issued his death sentence? Not so fast. They couldn't have done that if the reigning monarch of the Church, Pope Clement VIII, hadn't declared him a heretic. Pope Clement VIII has not been remembered for his clemency. So, whom should we try for the murder of Giordano Bruno? You might argue all of the above. But if you think about it, it was a system that brought about the murder of Bruno. It was a communication system that led to his demise. The human players in the system were merely the functional nodes to which information was addressed, nodes in which information was processed, and then emitted in altered form and addressed to multiple other nodes. That communication system, by the way, is still in operation today. It's called the Roman Catholic Church. When I started writing about physical immortality back in the 70s of the past century, I was certain that genes were involved in programming the lifespan of at least some organisms. But I was woefully ignorant of the complexity of the information systems networks in which genes operate. In fact, the notion of living systems as intricate communication and signaling systems had not even occurred to me. I had not then heard of Ludwig von Bertalanffy's general system theory, which took a holistic approach to living organisms, and it tried to understand them as complex functioning signaling systems. As you can see from the diagram beside me, even a small part of the network of genes that relate to longevity is complicated and difficult to understand really well. This chart is a diagram of just some lifespan influencing genes that act by affecting the rate at which DNA damage occurs and the rate at which DNA repair can take place. Now, DNA repair is crucial in extending lifespan because DNA, containing our genetic instructions, is continuously being damaged by all sorts of factors: radiation, toxins and poisons, free radicals and other oxidants, as well as just plain chemical bungling in the cell. If the errors aren't corrected, the recipes in the cookbook of life become garbled to the point where all we can produce is gunk. Just to give you a sense of how this chart relates to this program, notice the gene age 1 toward the upper left-hand side of the chart. That's the gene discovered by Michael Klaas around 1983 and shown by Thomas Johnson later to be able to increase the lifespan of C. elegans by up to 65%. It was later shown to encode the catalytic subunit of class 1 phosphatidylinositol 3 kinase, otherwise known as PI3K. Don't worry, I'll chew that mouthful more thoroughly again in a later program in this series. Subsequent modifications of this gene were able to extend maximum lifespan tenfold. I've already mentioned that. Applied to humans, we'd be living over 700 years. Not quite Methuselah's, but stay tuned. At the top center of the chart is the gene DAF2. Now you may remember that's the one that Cynthia Kenyon showed could double worm lifespans when mutated in a particular way. Applied to you and me, that would mean a lifespan of about 150 years. At the top right side of the chart, you see the gene SIR2 encoding a histone deacetylase. Histone deacetylase is an enzyme that regulates gene expression in a more or less permanent way, and it is implicated in the development of many cancers. Most exciting, however, is that SIR2 and another deacetylase, RPD3, have been linked to the effects of caloric restriction. Now that's a fancy word for semi-starvation. And it's involved in the extension of longevity. Now I'll have a whole program on that amazing phenomenon. Finally, for this first program that deals with Methuselah's genes, we may note that the gene TERT in the lower left-hand corner of the chart, that is a gene coding for telomerase reverse transcriptase, an enzyme you'll be telling all your friends about after seeing my next program. It will be entirely devoted to the subject of telomeres and telomerase reverse transcriptase and what I've been doing about them. That's all for today. Tune in next time to learn about telomeres in the aglets of Methuselah's genes. You might want to look up aglet in your dictionary if you don't already know what aglets are. For American Atheists and American Atheist Press, I'm Frank Zindler.